try to track as much as you can in terms of your health parameters, be it your heart rate, your blood biomarkers. If you can, DNA is obviously, as we talked about here, very important to give you a lot of biological context. Of course, if I were to choose one intervention, it would be vigorous physical activity.
Welcome to Longevity by Design, a podcast designed to give individuals access to the leading scientific information in the field of longevity. The ability to add years to your life and life to your years needs no opinion. Join us as we ask science to take the wheel. In each episode, Dr. Gil Blander joins a co-host and an industry expert in the field of longevity, shining a light and getting the answers to the key question: how can we live a longer, healthier life?
Hi everyone, welcome back to another episode of Longevity by Design. I'm your host, Dr. Gil Blander. In this episode, I interview Dr. Bartek Nogal, who is a principal scientist here at InsideTracker. Dr. Nogal leads the genomic team at InsideTracker, and his team recently wrapped up a very exciting project. They added 10 new healthspan-focused DNA scores into the InsideTracker platform. Bartek is an expert in genetics. This episode goes deep into explaining how to improve your health span using DNA insight. We talk about things like bone mineral density, epigenetic age, acceleration, cognitive decline, risk of elevated ApoB, grip strength, and its relationship to longevity, and more. Knowing your genetic predisposition to certain traits can help you fine-tune your wellness plan, and Dr. Nogal and I explain how to do so in this episode. I love this conversation as it's always fascinating to talk about actionable ways to measure and improve your health, especially via genetics, which historically was considered to be less actionable. If you enjoyed the Longevity by Design podcast, support us by rating and reviewing it. Thank you so much. Hello, I'm Gil Blander. Welcome to Longevity by Design, how to live a longer, healthier life. We are produced by InsideTracker, your science-based guide to optimizing your body from the inside out. Our guest today is Dr. Bartek Nogal. Bartek holds a bachelor and master degree in biological engineering from Cornell University. Following the completion of his master degree, Bartek worked in the biotech industry for 9 years, contributing as a process development engineer. He purchased a PhD in the Structural and Computational Biology program at Scripps Research. During his doctoral studies, Bartek employed a biophysical approach utilizing cryo-electron microscopy to understand the mysteries of naturalizing antibody response to HIV vaccine immunogens. His research resulted in numerous peer-reviewed publications, including contribution to the journal Cell. Furthermore, his doctoral thesis work received recognition and made the cover of the March issue of Cell report. Bartek joined the InsideTracker team as a scientist 5 years ago and worked in many different positions in the science team, and in the last several years lead the genetic genomic team. Welcome to the show, Bartek.
Glad to be here, Gil.
Excellent. So, Bartek, before we start and dive into genomic and genetics, I would like to learn more about your background and specifically what led you to become a scientist.
Yeah, I think it's a pretty simple story. I grew up just being really interested in the life around me. As a kid, I surrounded myself with animals. I spent a bunch of my summers just hanging out in pet stores and helping store owners with tropical fish and stuff like that. I had, multiple types of animals at home at one point. I was actually breeding hamsters. I had over 40 hamsters at home. I had guinea pigs, tortoises, anything I could catch, like salamanders, newts, any injured bird I would bring into the house. And so I was on track to what I thought would be a career in veterinary medicine. And I kind of kept that dream alive until I started in my undergrad program. And that's actually was the main reason why I sought out Cornell, because it has a really good world-renowned veterinary school. But really quickly into my freshman year, I started discovering the molecular side of life, bioengineering, molecular medicine, and that really piqued my interest then. And I made the decision to actually transition into those finer details of biology as opposed to veterinary medicine. That's sort of the story.
It's a good story, a very interesting story. And another point that I think that it will be interesting for our audience to know is you are part of the InsideTracker science team. So I think that it will be interesting for the audience to know why have you decided to move from the academia to the industry, and why have you decided to join InsideTracker?
Yeah. So I really love the idea of application of knowledge into something that's usable, and actually can make a change. In the case of InsideTracker, InsideTracker always really aligned with my lifestyle. So it was a natural transition and kind of a merge, if you will, of the kind of life that I was living um, with the type of work that InsideTracker was pursuing. So it was a kind of a natural marriage of what I was doing anyway, life.
Cool, thank you. And just a side story, I remember when Bartek actually joined us as a writer. We were looking for someone to write a blog about SHBG, sex hormone binding globulin. And I got introduced to Bartek by one of our team members. And he wrote an amazing blog. It was like 50 pages, it was almost a book. And we had to make it shorter and shorter. I don't remember, Bartek, if you remember that. And then I was really impressed by the blog that he wrote, and I said, "Hey, do you want to write another blog?" And he said, "No, I'm not for writing a blog. I want to do a real science." So he started to join us, and it was during his graduate study, and we got really impressed by his capabilities, and together we decided that he should join us full time. And since then, everything is history. So first, thank you so much for joining the team, Bartek.
Thank you. It's been a fun journey.
But we are here not to talk about InsideTracker and about your history or my history. It's more about genetics and DNA. So I would like to start with the basic and maybe even the super basic. So can you define what is DNA and what is its role and what is it doing, and how can audience that is not a PhD understand the basic?
Okay. Yeah. So DNA is a biological polymer that happens to be the code that kind of determines who we are as people. And so it essentially contains information to make the functional units of makes a human, and an animal, any kind of animal or plant, that kind of entity. So the central dogma of biology is DNA makes this other molecule called RNA. And then from RNA, we translate it into protein, and proteins are the business end of the dogma, where they are actually the enzymes and the structure building blocks of what makes your skin, what makes your organs, what makes your muscle tissue, and everything else that composes life. So yeah, that's sort of the basics of DNA.
Thank you. And just for a reference, if you look at the DNA of, let's say, a mice versus a human, how much of it is similar and how much of that is difference between us and the mice?
So I actually don't know the exact overlap, but I can tell you something maybe closer to us, where if you look at a chimpanzee and a human, the overlap is about 98%. So a mouse it's probably something downward from there, but it's probably would still surprise the listeners that how high it actually is, even if you compare it to, something simple. Yeah.
Yeah, so as much as I remember, it's around 95%, and you said chimpanzee is 98%. So for the audience, think about it. Only 2% of our DNA is different between us and chimpanzee, and still we are so different. So the difference that we see are localized in only 2% of the genome, which is really fascinating. And maybe that's a good segue to talk about SNP or single nucleotide polymorphism. Maybe you should explain that and what does it mean and what can you do with that?
Yeah, so SNPs are sort of the go-to variation for uh, determining polygenic predispositions to particular health traits. And they are the point mutations that develop over time in population, usually as an adaptation to the environment. So depending on where a population lives or where it migrates, it might develop SNPs to better absorb something from the soil because maybe that soil is not rich enough in some minerals. So you have to increase your capacity to enrich for that mineral from foodstuffs. Or maybe you're in an environment where you transitioned out of say sub-Saharan Africa back in the day, and now you're in somewhere in Scandinavia and you get half the amount of sunlight or less, and now your skin is not able to produce as much vitamin D. So over millennia, over generations, it behooves that population to evolve to be able to adapt to the environment to prevent fractures and improve bone mineral density to be able to absorb efficiently from the vitamin, the sun rays in order to be able to produce vitamin D in the skin in an environment that doesn't have as much sunlight. And there's, you know, thousands or actually probably, millions of examples if you want to talk about SNPs of those types of adaptations.
So we have in our genome millions of SNPs that basically that's what make Bartek different than Gil and Bartek different from any other human. And then the question is, should we look at a specific SNP or should we look at them in the aggregate and basically make score? And if we're making score, how do we do that?
Yeah. We can break that question up into two, let's say a two level on a high level, two parts. There are so-called monogenic or Mendelian types of conditions or syndromes or diseases where a single gene variant can have a very strong effect. It's so-called very penetrant. So something like Tay-Sachs or Huntington's disease, those are the types of things that we screen newborns for, even prenatally. So those single mutations can be enough and are enough to diagnose because those are diagnostic. When we talk about health traits, which is what InsideTracker furnishes for users, we are talking about complex traits that are best assessed by these so-called polygenic scores, where in a genome-wide fashion, we survey SNPs that are statistically significant in their association with a particular trait, like, for example, LDL level. So the more of those SNPs you have, typically, the more your predictive ability goes up for a particular polygenic score. So the state of the art right now for complex, particularly predispositions to health traits, are polygenic score and not single gene variants, just because the technology is such that it's relatively inexpensive to genotype, or even nowadays do whole genome sequencing to be able to calculate those polygenic scores for people. And the way a polygenic score is calculated, it's a weighted sum. Let's say you have, we can use one of the new scores, say bone mineral density. That's about 900 locations in the DNA that associate with having a lower or higher bone mineral density. And those were found in a so-called genome-wide association study. And from there, some of the statistical inferences that are made, you get an effect size that allows you to weight any particular location on the DNA in terms of how much effect it has on the trait, in this case, that being bone mineral density. And you create a weighted sum. It's pretty simple of those effect sizes. And you come up with a single value that then can be used to compare you to the population. And in that sense, it gives you context where you are on the spectrum for risk for lower bone mineral density, for example.
Okay, so let me see if I understood you correctly. What you're saying is the following. The polygenic scores is basically scientists make an experiment, and let's take the LDL, as you mentioned before. So they can look at a big population, let's say 10,000 people, and they know the level of the LDL for all of them. And then they are trying to take, let's say, the top 10% and the bottom 10% and try to see what are the single nucleotide polymorphisms that are different between them. And then based on that, they calculate a score and come saying, well, those 50 or 100 or 200 are the single nucleotide polymorphism that differentiate between those two groups. And then they come say, if someone have this kind of allele of those SNPs, they are a high risk for LDL versus the other one is low risk. That's a fair assessment, or am I made any mistakes?
I think what you described is more of a classic GWAS for a disease, a presence or absence of disease. When we're looking at LDL as a continuous variable, you basically say you have, say the UK Biobank, you say you have 500,000 people. For each person, you have their DNA array data, you have their LDL measurement, and you run a linear regression to find out which of the genetic variants on those arrays for each person associates with LDL. And from there, you calculate, you basically create the so-called Manhattan plot, and it tells you which SNPs are important. And from those SNPs, you're able to do a bunch of statistical manipulation, only take out the ones that are not redundant. And from there, you can calculate a score. And then, yes, when you have the distribution for the score, you can create percentiles to be able to tell people, say the people who are in the lowest 10%, they're at low risk. People are at the highest 10%, they're at higher risk.
Good. Thank you for the explanation. I think that it's a very important point for our audience to understand how we calculate those polygenic scores. So now my next question is, those polygenic scores, what percentage of your, let's say, the level of LDL or your bone mineral density they explain versus other things like environment and behavior and so on? Can you provide a few examples just for users or listeners to understand what is the ballpark of the explanation?
Yeah. So maybe what I use is basically I'll try to explain heritability, because essentially that is what we're talking about. So the concept of heritability can be a little bit tricky. So when you have something like ApoB, so for example, if we say that ApoB is 50% heritable, it does not mean that 50% of Gil's ApoB is due to genetics. It means that if we look at say 10 people or a different number of people, and we measure all of their ApoB levels, the variation that we find out from, let's say the average for those 10 people in ApoB is 100 milligrams per deciliter. And let's say the variance is plus minus 5. So if we say that heritability is 50%, we can say that 50% of that variation is due to genetics. So again, it doesn't mean that on an individual level, 50% of your ApoB or LDL level comes from genetics. It means it's a population metric that measured at a particular population at a specific time is some number. And yeah, it can change over time depending on how we measure it and what technology we use. So it's a ballpark. It's not an absolute value. I'll give you an example. For example, I kind of like to use this. Suppose we have a polygenic score for body mass index, BMI. So how heavy you are. And we have this polygenic score in the US population where food is freely available. People have access to as much food as they want, pretty much, because you can, let's say not food, but let's say calories, right? Now you have the same polygenic score, and that's for the sake of argument, let's say the genetic background of another, say, third world country is the same as the population we're measuring in the US. but now this third world country is going through a famine. Now the genetics of these people are the same, but if you run those scores on them and you measure their weight, you're not going to get the same heritability. So because the environment isn't allowing for that heritability to express itself. So that sort of kind of gives you an example, an extreme example of how what heritability is and is not, right? Same thing with, for example, like a solid psychological variable like IQ. So if IQ is 50% heritable, it doesn't mean if you score 160, it doesn't mean that 80% of 80 of those points are due to genes. It just means that if you get 10 people, the variation, whatever variation is within their IQ, 50% of that is genetic.
Yeah, so it's looking at population. It's not a fit for Bartek or Gil. It's for population, but it gives us provide the ballpark. So you spoke about ApoB and you said that it can be up to 50%, but that's an extreme, correct? Like let's say
so those heritability estimates are based on the broad-based heritability, where you're able to take a set of monozygotic and dizygotic twins, track them over time, and then adjust for confounders and see using some structure equation modeling, you can figure out what the heritability is. That's the ceiling, because you're accounting for all of the genetics without measuring any genetics, because you're looking at you have a controlled experiment, a controlled natural experiment. When you can also calculate heritability from the latest technology through array technology, but those arrays only capture, for the most part, common variants. They don't capture anything that's super rare, like say less than 1% frequency for a particular allele. And so when you're measuring heritability or the variance explained using that kind of technology, you only get a portion of the true heritability. And so the other kind of the gap is called the question of missing heritability. And that's still kind of a controversial topic where most of that missing heritability is coming from. But in general, people think that it's due to these rare causal large effect size variants that would help close the gap. Once we're able to whole genome sequence millions of people, which is very expensive. That's why we haven't done it yet. At that point, in theory, we would be approaching for ApoB the same heritability that we can see with like a twin-based study. Now, when we have a polygenic score for something like ApoB, and we have at InsideTracker, we have the benefit of actually having our users, both their DNA data and their blood data, we can run a linear regression on that and get an estimate of the variance explained by the polygenic score. And because ApoB is known to be, highly heritable, we do in fact reproduce qualitatively that we actually do see double-digit explained variance in the ApoB. And to give you some context, so when our score right now explains about 14% of the variance in our users' ApoB. So in plain English, when we look at the bottom 10% of the distribution for the polygenic score, our average users' ApoB levels is about 77 mg per deciliter. Now, when we compare that to the upper 10% for ApoB, so people at highest risk of ApoB levels, their ApoB levels are closer to 110 mg per deciliter. So that gives you an idea what 14% heritability can do. And so I don't have the numbers with me, but I'm pretty sure that 80% mg per deciliter is way below the 50th percentile for ApoB levels, and 110 is up there in a generally healthy population in terms of what ApoB where it falls.
Excellent. And if we will stay with ApoB, for example, the question is, so what? So I know that I have a high risk for a high genetic risk, let's say, for high ApoB. What practical decision that can support? What can I do with this data?
Yeah. So first of all, anything I say from now on is not medical advice. It's just inferences from the latest scientific literature that I am personally familiar with. And I'm also going to extrapolate from where we have more data, which is LDL, which is very closely tied. The polygenic risk score for ApoB and LDL are very closely tied. If you plot them together, they're essentially, a very high correlation. So when you have a high polygenic risk for ApoB, now, are you saying that with also having information about your blood levels, or?
Yeah. Let's assume that yes, I have the blood data of my ApoB and I have my DNA risk. What can I do with that?
Yeah. So in general, it gives you an idea of either how good you're doing in terms of your lifestyle. So if you have a high risk, but your ApoB level is, low, then you're probably doing something really, really good in terms of your lifestyle. If they match, then you're basically living up to your genetic potential. But there's also, and then this is where I was alluding to, there's also an element of polygenic score that doesn't just tell us what the level of the biomarker is. There is some inherent, very deep biological insights that go far and beyond what its prediction of the blood biomarker. I'll give you an example with LDL that's been published in the literature. And while this hasn't been necessarily reproduced explicitly with ApoB, but it's likely that's very similar because the biological pathways behind ApoB and LDL could be similar in terms of their effect on cardiovascular disease. So when you look at individuals who are at low risk for LDL levels versus medium risk for LDL, genetic risk for LDL levels, and those who are high, a person who is, has their LDL is 110 mg per deciliter. 110 is below the clinical threshold for, right? I'm just trying to use a number that makes sense. And they are at high risk. Their risk of cardiovascular events, so the odds ratio of having a cardiovascular event at 110 is much, much higher than a person who has an LDL of the same 110 and has low genetic risk. So this is really the definition of precision health medicine, right? Because you're not just looking at the polygenic score to tell you where your LDL should fall, but even if you have a polygenic risk score that's high for LDL, but your LDL is low, or not, it's the same as a person with a high risk. That risk of a cardiovascular event is different if you have the polygenic background.
So I'm trying to summarize it and actually say it in my word. What you are saying is you can look at the real number of the LDL and the risk of LDL as a different indication. So a person that have currently, let's say, a LDL score of 110, but he have a high risk for having high LDL, basically have two indications. One is the genetic, his potential is to have high LDL, but now his LDL is okay, which is good. But he's still in a higher risk to have higher LDL than someone that have the same level of LDL blood level, but lower risk of LDL, meaning that blood and genetic together are much more powerful than having only the blood data or the DNA data alone. Is it fair what I said so far?
Yeah, I think you got the concept, but what I mean, basically LDL biomarker levels for LDL with the polygenic risk background are not created equal for people with different genetic risk. And what we're talking about, the risk of cardiovascular events. So regardless of, basically to summarize, regardless of LDL levels to a point, if your polygenic risk for higher LDL is high, you always will have a higher risk for cardiovascular events. So that's where you kind of have to, even if you modify, this is where even if you're able to modify to a point your LDL levels via lifestyle, but your genetic risk is high, it would still, and this goes maybe for ApoB as well, it would still behoove you to have a serious conversation with your physician about the other thing that's not measurable yet, and that's future cardiovascular events. Because like I said, these polygenic scores, especially for lipids, they don't just capture the math behind what calculates an LDL. They capture a lot of biology that relates to epithelial, health, and all kinds of, resistance to stress, different cellular differentiation, things that relate to health span overall, and in particular, heart health.
Yeah, but it's that's a fair point, Bartek. But I'm still trying to communicate the following message, and I would like to be sure that you agree
Yeah.
with me that someone that have higher PGS score for LDL should take his real LDL level, the blood level, more serious than someone that have a lower risk, because he's
Yes.
basically in higher risk, and then
Yeah.
he should maintain his LDL as low as possible because he already have
That's
a
it.
high risk.
I would agree
Yeah.
with that. You should be, regardless of the number, you should be as aggressive as
Yeah.
possible if
Yeah.
you have that. Yeah. And
Okay.
actually,
And
I was going to say, if we're talking about lipid levels, familial hypercholesterolemia, that's not something we look at in InsideTracker because it's a more of a monogenic, highly kind of diagnostic trait. But it also shows, so familiar hypercholesterolemia is not, it's very penetrant, meaning if you have the genetics behind it, you're more likely to have super high cholesterol early on, but it's not 100% penetrant. And what research is actually uncovering is some of that missing penetrance, it can be explained by polygenic scores. And the same kind of polygenic scores that you can measure in healthy people, meaning people who have the FH genes and should have high cholesterol don't, they could have a very low polygenic score to help protect them from it penetrating, meaning expressing in the form of the phenotype of having very, very high LDL cholesterol.
excellent. So I think that we spent some time, but I think that we at least try to explain. And again, it's very complex. So hopefully we have been clear enough. We try to explain the combination of blood and DNA together and why it's better to measure both and not only blood or DNA, which is great. What I would like to discuss right now is what other genetic score InsideTracker is currently test other than the DNA score for high level of LDL or high level of glucose. What are other traits that we are testing and why?
So right now we're looking at a bunch of polygenic scores for most of our blood biomarkers, not all of them. We have a lot more in the pipeline, and it's really, really soon we will be unfolding between 9 and 10 brand new scores that are specifically related to health span traits. And those are things that are very important for healthy aging and increasing the disease-free portion of a person's lifespan.
Excellent.
So,
And we will spend the majority of this discussion today about those 10, which I'm
Okay.
super excited
good.
about. And I assume that you are also super excited about that. But
Absolutely.
what is, if I will ask you, and I will ask you, to say which one of them is the one that Bartek is most excited about? Which one is it?
Oh, I would say it would be the one where we can interrogate using our own internal data, and that would be ApoB, because we
Okay.
do have ApoB measured, so we're able to actually validate it internally and really vouch for its really good predictive ability. But all the other ones I'm also very excited about, just because of the research that went into coming up with these scores. One of the criteria was that all those traits have a good proportion of heritability, such that when we're giving you a rating for a genetic risk, it's actually meaningful because genetics is contributing to that trait.
So can you, Bartek, elaborate a bit about that? How many study do you need in order to decide that we are adding this score? What was the N number, the minimum N number? What should be the effect size? If you can discuss a bit about that, that would be, I think, very beneficial.
Sure. Yeah. So the beauty of the human genomics field is that data is very freely available, and it's shared among not just academics, but it's freely available on different repositories. There are multiple different repositories. A lot of them tap into the largest study worldwide right now, which is the UK Biobank, which is, a generally healthy, population, of people between in their 40s to 70s. And, that research study basically went ham on, phenotyping these people in many times in longitudinally, and, when researchers apply to look at that study, they publish their genome-wide, study findings. And then, people like ourselves can go into these repositories, look at the GWAS statistics, GWAS meaning genome-wide association study, and we're able to, extract the most significant variants for a particular trait. So, when we look at, the discovery GWAS behind the polygenic score, we look at, a study size of at least, in the tens of thousands of people. For the most part, it's in the hundreds of thousands of people, but some of the traits that are healthspan traits are maybe, not as well represented. So, oftentimes, sometimes we'll find studies that are maybe more like 70,000 people. And we also look for meta-analyses, which is a way to combine multiple GWAS to increase your sample size, and then also to increase your ability to discover more variants with a larger sample size. So that's kind of the process. We look at, different GWAS summary statistic repositories, and we, extract the most significant variants for particular traits, based on those, repositories. And when it is a trait such as ApoB or LDL or hemoglobin A1C or fasting glucose, these are traits that we measure within our population. These are polygenic scores that we can internally validate. So by that, I mean, we can take our users' anonymous DNA, match them to their anonymous blood data, and then, run a so-called linear regression to see if that polygenic score that we're trying to discover is actually predicting, the blood biomarker in our user. And if it's not, we iterate. We go to the next score, or we look at different variants until we get an optimal, prediction for where we can get. Because, there is a so-called concept of transferability of scores. Just because a GWAS was run in a particular population and it explained, a certain proportion of variance in that population where it was discovered, it doesn't mean that if you transfer it to, a different set of people, it doesn't mean that we'll be as good at predicting. And typically it's a little different. So that's how a lot of research goes into it to make sure that, we're generating insights that are actually valid and insightful for a user and more likely to be predictive of where people are.
As a Longevity by Design podcast listener, you understand the value of improving your health for today and for all the years ahead. And if you want to live your healthiest, longest life possible, you need to understand what's going on inside. At InsideTracker, we take a personalized approach to health span optimization that eliminates guesswork from your wellness plan. InsideTracker analyzes blood biomarker and DNA data, along with physiomarker data from fitness trackers like Oura Ring, to deliver personalized food, supplement, lifestyle, and exercise recommendations that allow you to take control and improve your health span. And for a limited time, Longevity by Design listeners can get 20% off at the InsideTracker store. So if you're ready to receive a personal health analysis and data-driven wellness plan to optimize your body for the long haul, then it's time to start inside. Visit InsideTracker.com/podcast to get started today. That's InsideTracker.com/podcast to get started today.
Got it. That's good. And just to explain to our audience, how many SNPs you have in, I would say, in average or a normal or example, because it's very dramatically. But let's take the ApoB that you discussed before. How many SNPs are included in this score of ApoB?
Yeah, so the score we converged on, we looked at about maybe 3 or 4 different scores, and they were between, say, 100 to maybe in the hundreds of thousands of SNPs. But the most manageable and the most predictive score that turned out to be most predictive in our population was a score that's about 17,000 genetic variants. Yeah. So basically when a user uploads their DNA to InsideTracker. We look at their array. We have this so-called imputation algorithm where we're able to infer missing links, and we're able to come up with those 17,000 for that user, and then use those variants to do the summed average weighted by the effect size for that user, so they get a number and a risk score that they see in the app.
So the non-scientific listener, we are looking at thousands, if not tens of thousands, different locations in the DNA that they are responsible for your risk to have a low or high ApoB. So it's a very big score with a lot of information that allow us a pretty accuracy to predict how likely you are to have a ApoB just based on your DNA, not looking at the environment and not looking at the other thing, which is maybe was a magic, I don't know, 5 or 10 years ago. Today is doable, and we are doing it. And we are doing it based on a very strong science that was published in the best scientific journals and was done on a very good database that called the UK Biobank. So I think that that's a very good background information, Bartek, and now, with your permission, I would like to go specifically for a specific scores, but maybe
Let's do it.
before that, maybe either you or myself can mention just what are the scores that we are currently adding for what we call DNA healthspan scores, and maybe why in a sentence or two for each of them.
Yeah, so you would think I have them memorized. So I do have a list here. Let me start with, they were identified based on a few criteria. One of them being, is it related to healthspan? And one of the other ones, again, are they actually heritable? So we wouldn't pick a score that's, we wouldn't pick a trait that's, I can't think of one, but one that's healthspan related, but there's no evidence that it's explained by genetics. It just wouldn't make sense. We could come up with something, but it would not be very predictive in our users. So I'm going to start with visceral fat. So visceral fat is a type of metabolically unhealthy fat that tends to surround the organs. It tends to be correlated with having a bigger waistline. It's actually an independent predictor of metabolic disease, regardless of any other blood biomarkers. And it's also kind of a silent, if you think about it, it could be a silent killer. If it's not something you go and you get measured, unless you get like a DEXA scan where you want to look at your bone mineral density, your body fat percentage. I think that's like a secondary measure that you can derive from there. But most people, and including myself, I don't know what my visceral fat is. I'm relatively lean, but I actually happen to have a high risk for increased visceral fat. So that's making me actually think like maybe I should invest $100 and go get a DEXA scan, because I know my lifestyle is pretty good. And typically I look healthy, but what if that risk, I actually haven't modified it enough and I have to think about it. So visceral fat is a nice one because again, there's not a readily available biomarker for it. You have to go out of your way to see it.
Yeah, and Bartek, let's say just a comment about visceral fat. So I think that's a great example, and I done a DEXA scan a few months ago, and I was shocked that I have a high visceral fat, and I didn't know about it. By the way, I don't know yet what is my risk score for visceral fat because we haven't released yet the DNA score. But that's a great example of something that you can test the DNA and see if you have high risk. And as you said, then if you have high risk, go and do a DEXA scan. And if you have high visceral fat, then there are a lot of intervention, what you can do in order to manage this high risk for visceral fat and maintain it in a low level. Okay, what is next?
Yeah, so just to speak to that a little bit. So the way we think about these genetic insight and all the ones that will come up in the coming years is basically a roadmap. So what you just described, a road in your healthspan journey that you can explore that you didn't know about before, that leads you to a better healthspan somewhere down the road. And knowing your predisposition, whether it's average or high, especially if it's high, it's at least starts a thinking process toward in that direction, toward more informed optimization of your wellness. Another one, let's say muscle weakness. So that's an age related muscle weakness. That's going to be a score that's a bit of a late onset. So this is a score based on an elderly population above 60 or so. But again, polygenic score are something that can be determined at birth. It's not something that changes over time. So if you know your predisposition to muscle weakness at say 45, you're not exactly at that point where it might be expressing itself. But if you're at a higher risk for having muscle weakness in older age, you might think about having an increased amount of resistance training or any type of muscular endurance or strengthening exercises to make sure that as you age, you maintain your muscle strength and reduce your risk of frailty and falls and being able to maintain your balance as you age. Those are not things that we think about, in our 30s and 40s and maybe even 50s. But as we age, if we do have the genes, and I should say the genetics behind muscle weakness in old age are distinct from the genetics of continuous muscle strength in overall in an entire population. So it's not like if you score high on another score that we have, which is hand grip strength. That's a new score that we also have. If you score high on hand grip strength, which tells you that maybe you have better genetics for having stronger muscles, that doesn't mean that as you age, you can't score differently on the other score because those genetics are distinct. Okay.
Yeah.
Any questions?
Sorry. Yeah. A comment about that. So muscle strength is extremely important when you get older because I'm sure that you know that, Bartek, but hopefully our audience knows that the majority of deaths of old age is basically that the old person fall, break his hip, and then or other bones. And then it's amazing that the survival curve of a year after breaking your hip is maybe more than 50% of the population that break the hip at the age of 65 plus are not surviving a year after. And,
that's an incredible statistic. It blows my mind.
It's crazy.
Yeah.
It's crazy. And again, if you know your risk and you know that, you can basically build your muscle. And if you build your muscle, first you have better balance, so less likely that you will fall if you lose your balance because you have better balance. And second, if you fall, then you have more muscle to protect your bone, so you have a lower risk to break the bone. And if you break the bone, still you have better a muscle to recover. So that's something that is no brainer. If you know that, it's the best sign that you have. Start going to the gym and build your muscle. It's not a rocket science. So it is an amazing, and I will strongly recommend everyone to test and see whether they have higher risk.
And just to clarify, while the two traits are not entirely independent, they're intertwined, meaning muscle mass and strength, they tend to go hand in hand. But if you were to choose one that's more important for health span and longevity, it would be strength. So the person who is light on their feet and is strong per pound of their body weight, so meaning they're strong for their body weight, will probably do better than somebody who's very muscular but not as strong for their size. So sarcopenia is another danger of aging, and that's a kind of a complex phenotype that includes, frailty, loss of muscle mass and strength, as well.
Excellent. So what other scores do you like in our?
So maybe along a similar vein as in bone mineral density, because bone mineral density is something it's very known to be a high risk factor for fractures. And so if you go with that example of elderly person falling, if they have a higher bone mineral density and they're not osteoporotic, they're less likely to fracture something. And how do you build bone mineral density? Physical activity, loading the bone throughout your lifespan, and resistance exercise. So that's a very important one. And if somebody scores, and say they're in their 40s and 50s, and they score low on bone mineral density, that's another one that can help propel a person to go get a DEXA scan, because they may not be falling yet because they're young, and there's a lot of compensatory mechanisms for preventing falls, and you still have good, your brain has good reaction time, good processing speed. Once that's not there and you trip and you're not able to, essentially control your movement, and and you fall, and your BMD, bone mineral density, is low, you're more likely to fracture something and then have serious complications. And
Yeah, absolutely. And again, it's going to the DEXA. So the DEXA scan is an amazing scan that you can find so much information by lying on a machine for 5 minutes and receive a very low radiation, which is something that I also highly recommend to everyone that want to live a longer, better. It's also not expensive. I've done it, and it's cost me like, I don't know, $100 or something like that. It's not crazy. A few visits to Starbucks and you cover or you save a few visits to Starbucks and you already get the information. And it's also like blood test. It's very important to have a reference. So like we're talking about blood tests, it's very good for you to have the baseline and then to have the follow-up. And then my bone mineral density went up or went down. My visceral fat went up or down. My muscle tone went up or down. That's all of that you can get from DEXA. So do it as early as you can, and then continue to do it every few years, and you will have a very good reference to see whether you are in the right trend or in the wrong trend.
I think I'm sold, Gil. I'm going to make an appointment today.
Okay. Great.
Maybe we'll switch gears and go to Chronotype now. Specifically, we now have a polygenic score to help determine your genetic predisposition to being a morning person. So a lot of epidemiological studies have identified morningness, meaning the people who are these so-called morning larks. They get up early, get their stuff done early, perhaps exercise early. They have more healthful behaviors, including less predisposition to some metabolic conditions like type 2 diabetes and such, but also may choose healthier foods. Whatever the mechanism behind that is, it hasn't been really elucidated. But in general, morningness tends to align with more kind of healthy traits than being an evening person. And there's some controversy, not necessarily controversy, but there is some conversation around whether it's the morningness or is it misalignment. And those are kind of tricky things to answer, but the large body of evidence points to being a morning person, aligning more with the natural circadian rhythm of the sun is more helpful for your health span than, perhaps being a shift worker or a night shift worker or something. While your body will entrain on any circadian rhythm you throw on it, and especially if you do it consistently, your body will adjust, your enzymes, your clock genes will adjust, but it doesn't mean that in the longer term, you won't be more predisposed to type 2 diabetes or obesity, or, again, some of those behaviors that may predispose you to eating more, processed foods or snacking more, etc.
Yeah, I will add to that on top of all the health information that I completely agree with you, there is also some behavioral information that is important to know. For example, I'm a morning person, so I'm not making an important decision at 9 p.m. or I'm not studying or trying to learn something at 9 p.m. I know that it's the time that is better for me to do it, I don't know, 7 to 10 a.m. So that's the time that I'm dedicating more to study or to learn or to make an important decision. So I think that knowing whether you're a morning person or evening person can allow you to know what is the best time of the day for you to make an important decision or to learn a new subject or to focus on the thing that you really want to focus on. So I think that it's another a value point that you can use when you know whether you're a morning person or an evening person.
Yeah, on that note, there's actually some, say, moderately weak to moderate evidence that the same goes for exercise and endurance training. So endurance athletes who are morning, they're more likely to get more performance benefits if they actually align with their morning training versus, in the evening. And same thing for people who are evening types. There may be a different dynamic there where they may actually have less benefit by trying to train in the morning. But I don't remember there's a little bit of evidence there that aligning your training with your chronotype can be beneficial.
Yeah, that's fascinating. I didn't know that. Thank you for letting me know. That's exciting.
Again, not really strong evidence, but it would have to be determined on a N of 1 basis for every person.
Yeah. No, but it's good to know. So what is the next one?
Well, maybe we can transition to maybe longevity.
Okay.
So we have a score that's basically was determined, looked at about 1 million individuals, and look at the continuous lifespan in these individuals. And there was a score, there were variants that were identified that explain some pathways behind cellular differentiation, resistance to stress. Some of the classic kind of longevity genes like ApoE were identified there as well. But then there was an independent study who actually took some of those variants, and in an independent cohort, they used those variants in the form of a polygenic score, and they were able to discriminate between centenarians and normal old people. And they also were able to identify younger individuals who would live longer. And so that was a really nice framework for us to start with, to start building a longevity score, because the validation was already performed for us. So we weren't tapping into a discovery GWAS and hoping, since we can't measure longevity in our users, we were able to use another study that used the exact same variants from the other study to have a validated score that explains something like if a user scores, say, in the lowest percentile, they may have a lifespan, not accounting for any environmental or lifestyle factors. Their baseline lifespan might be about 5 years, 3 to 5 years less than the person who scores at the very upper limit, the upper fifth percentile, which is very, very rare. So again, this would tell a person at baseline what your potential for longevity is, not accounting for your lifestyle. For example, you can modify that through lifestyle. So if you shouldn't be concerned if you score low, because if you're an InsideTracker user, you're most likely actually modifying your risk toward somewhere in the middle for that score.
Yeah, but is it fair to say that someone that score low for lifespan should work a bit harder than someone that score high for lifespan?
Yes.
Is it fair to say?
Yeah, it's fair to say, because like any other polygenic score, those score don't just capture the direct trait that we're trying to measure. They capture a lot of pathways and a lot of biology. To go back to chronotype, for example, a lot of the variants behind chronotype are in the clog genes like period 1, period 2, cry1, cry2, BMAL. There's actually, and those are proteins that are expressed heavily in the suprachiasmatic nucleus, which is a center in your brain that detects photons through your retina. There is like retinal ganglion cell variants that speaks to maybe there is some kind of genetic variants that modify the communication between the retina and the brain, how it detects light, and that can then determine sleep timing and chronotype. And so going back now to lifespan, again, all of these scores, they have a lot of biology behind them. So really, it's important to pay attention to where you score on those scores.
Yeah. So basically, again, so far, I want to summarize the intermediate summary to say those scores can give you a lot of information based on your genetic that you can act upon. And we already discussed a few of them and gave a few examples what you can do. So it's not like just a score, that it's not just information. It's also an information that you can act upon and basically be ready and prepare for your basically reach to your 100 years, hopefully, based on all of this information. You know what to focus on. If you have a low risk for low bone mineral density, you need to work on that. If you have a risk for high visceral fat like me, and it sounds like yourself, we know what to do in order to work on that. So we are giving you some information that you can act upon, and hopefully that will help you to beat your genetics and live better long.
Yeah, agreed.
Okay, good. So let's continue. What is next, Bartek?
So along sort of longevity traits, and so I'll take my time explaining this one. We have another score that tells you about your predisposition to epigenetic age acceleration. So there are some of your listeners probably have maybe on this podcast heard about epigenetic age, and that's
we discuss it. Yeah, we discussed it with in a few episodes. So yeah, but it will be good for you to repeat it because it's complex, even so that we discuss it a few times.
Yeah. So it's a measure of your so-called biological age. So we all know our chronological age. Most of us know our chronological age these days. We know when we were born. We know where the calendar is at this point. So that determines our chronological age. But as we know, people age at different rates, and there are various different ways, including InsideTracker's inner age, to measure your so-called biological age, which is sort of your inner age, like what your biology is doing, regardless of where you are on the timeline in terms of objective time. And another biological age measure that's been really well characterized by now is via the chemical modification of DNA. So methylation are these so-called methyl groups at different points, specific points on the DNA molecule that could change the way the DNA is read. And so what that can do when the DNA code is read, the methylation can either increase gene expression or lower gene expression, or structurally change the DNA in such a way that something different happens in the way your genes are expressed. And so those kind of modifications have been correlated to biological age, meaning lower occurrence of chronic diseases, for example. So a person who has an epigenetic age that is, say, a person is 35, and they say they're a smoker. They are a lifetime smoker. Even though they're 35, it's very likely that their biological age via methylation will be higher than 35. Because smoking is a really well-established risk factor for increasing, I would venture to say, any biological age metric out there, whether it's blood-based or DNA-based or methylation-based. We are not measuring methylation at InsideTracker. There are different methylation clocks that are represented in the literature. And there is one specific called intrinsic epigenetic age that's actually been found to be quite heritable. And so when something is heritable, that means you can look at the genetic variants that explain that heritability, and you can develop a genetic predisposition to increased or decreased epigenetic age acceleration. And so actually epigenetic age is actually at birth, it's about 100% heritable, because if you look at twins, they're going to be 100% same methylation. As you go further into a person's age, in your 40s or something, or as an adult, your epigenetic age is still around 40% heritable based on broad-based heritability in twin studies. So it's a very good candidate for having a polygenic score. And that is what InsideTracker has developed, a polygenic score for having a predisposition to increase or decrease epigenetic age acceleration. So it's a measure of using that specific way of measuring biological age. It's your predisposition for that particular age acceleration metric.
Okay.
I probably wasn't very clear, so maybe you can help me.
No. It was clear. So the bottom line is there are a few different kind of clock that can predict your longevity. Some are similar to what InsideTracker, and what we have is the inner age, so it's a blood-based clock. Other are epigenetic clock based on methylation. There are other that look at glycan age, so basically other modification. Some other that are looking at telomere lengths. And what Bartek and his team found is that there is a correlation between your genetic composition and a specific clock that is based of epigenetic clock. And that's what you are doing here. You are basically allowing a user in a way to predict what will be their epigenetic clock based on their genetics. That's fair to say?
Yeah, that's fair to say. Yeah. And if we were to, for some of your listeners who are InsideTracker users, if you were to put the three aging metrics we discussed just now, that being the lifespan score, the epigenetic age predisposition, and inner age in context, the lifespan score would be like your background, polygenic background for having a particular lifespan. Your epigenetic age predisposition is your predisposition to a specific aspect of aging that's highly modifiable. So if you compare the lifespan score to the epigenetic age score, the latter is a lot more modifiable by exercise, etc. And then when you look at inner age, which is based on blood biomarkers, that's a real-time readout of the direction you're going in terms of your biological age. So that we can kind of look at those three in that context.
Okay, what other scores do we have in our new scores?
Cognitive aging. So we have a polygenic score that estimates a person's predisposition to decline a cognitive ability through most of the lifespan. So that kind of score, again, you may not be worried about it when you're middle age or still in your 50s or something, but if you score higher, you might want to think about pay attention to some way to intervene in terms of some of the same healthspan interventions like exercise, oxygenating your brain through exercise, healthy diet, but also some cognitively demanding tasks like puzzles, or they have very specific apps to help prevent cognitive decline. If you score particularly high on that score, you may want to take some extra prevention steps, because mental health is incredibly important for healthspan, because what does it matter if you have low LDL or low ApoB if you're not experiencing life in a positive way?
Yeah, I agree. This one is huge, and we know that a lot of cognition-related diseases or brain-related diseases are correlated very strongly with your age. So you get older, you have a much higher chance to have them. So finding it early and intervening with the intervention that Bartek you just mentioned can make a lot of difference. So it's again very good to know early and know what to do.
And last and certainly not least, is we have a score for our women users, and that pertains to the propensity to have an earlier menopause. As many women and them kind of know from family history, menopause is quite heritable. So if a mom, a grandma had early menopause or late menopause, that kind of tends to run in families. So that's a clue that it's actually amenable to having a polygenic score and be able to try to predict a woman's predisposition. And that's an important one, because women who may go into menopause early tend to have and do have less estrogen exposure in their lifespan. And estrogen is very important for bone health, particularly to prevent osteoporosis and some other health conditions. So that's an important, especially for women who are still premenopausal. If they are scored as having a slightly higher elevated predisposition to have early menopause, it can help them start a conversation maybe with family members, their physician, to think about the types of things that are possible to do to help ensure, that, to ensure that you may help modify the slightly increased risk.
Okay, yeah, and that's definitely super important for our audience, especially the young audience that would like to know when they will have or predict when they will have menopause, and maybe it will help them with a family planning. So
That's
definitely
true too.
another good score to have in our a new panel, and yeah.
Yep, I would just add to that because we don't necessarily would be able. It's still any of these polygenic score. It's a probabilistic estimate. It's not something that will tell you you should have kids early. It's not at all diagnostic. So it's a conversation starter. If you do score, at a particular risk category, let's say that you should start thinking about looking into it.
And I think that Bartek, all the scores that you and your team added are amazing, and I think that everyone that would like to live better, longer, should test them and know, and based on that, know how to build this plan for reach 100, hopefully. So I think that that's an amazing addition to the InsideTracker solution. And I want to conclude our discussion by asking you a question about what is the tip that you would like to provide to our listener about how to live better, longer?
Ooh, I would say to the extent that you're able, try to track as much as you can in terms of your health parameters, be it your heart rate, your blood biomarkers. If you can, DNA is obviously, as we talked about here, very important to give you a lot of biological context. Of course, if I were to choose one intervention, it would be vigorous physical activity. Not to be forgotten is having a good amount of social support. Creating strong bonds in a community is very important. And yeah, if you can do those things, and obviously don't eat junk. Diet is also important. So those would be kind of the pillars of healthspan that, going forward, I think are important.
Yeah. So, Bartek, that was a fascinating discussion. Thank you so much for cleaning your calendar and spending an hour plus with us and discussing your work. And I'm really proud of what you and your team made. And I think that it's an amazing addition to the InsideTracker community to understand better their genetics, and part of our vision or mission to combine blood and DNA and physiological marker together, and the intersection between them. And very exciting. And thank you so much for joining us. And I hope to host you again in the next release of the new exciting features that you and your team will have. Thank you so much, Bartek.
Thanks, Gil. Looking forward to it. I had a blast talking to you, and thank you so much for the kind words.
Yeah, you're welcome.
Thanks for listening to Longevity by Design. Please subscribe to this podcast on Apple, Spotify, or YouTube. Longevity by Design is powered by InsideTracker, a personalized health optimization platform that helps people improve their lives by improving their bodies from the inside out, using personalized, science-backed recommendations for nutrition, supplements, and lifestyle changes. To learn more, visit InsideTracker.com/podcast.